¿Qué tal?
Hola Maribel,
¿Todo bien?
todo bien, todo bien.
Ah, espera, hola.
Hi all, how are you doing?
Hi.
Cuando
Shall
quieras,
we start?
Ismael.
Yeah,
Sí,
shall we
dale.
start?
Sí.
Well, let me introduce you, Thomas Darde. He is the founder of Silicium in France, a bioinformatics company specializing in transcriptomics and toxicogenomics for translational research and applied biomedical projects. His work mainly focuses on RNA-seq analytics, data integration, biomarker discovery, and reproducible pipelines, supporting both academic consortia and industry programs. He is involved in the hard-running Horizon Europe project, contributing to data analysis and the development of reusable analytic resources, as he is now the leader of the working package 4 of this project. So thank you for accepting our invitation, Thomas, and the floor is yours. Thank you.
Thank you, Ismail. Thank you for this introduction, and thank you for giving me the opportunity to talk about how we can use transcriptomics and more general multi-omics data integration for molecular signature detection in MASH and DILI in general. Uh, just to be brief, I'm nothing to say more about this introduction, just about the fact that I'm not an expert in the liver, not an expert on this domain. I'm mainly bioinformatician, biostatistician. That's why this talk will focus more on transcriptomics, or we use the transcriptomics in the DILI and MASH framework, and how this data could be used coupled with other omics technology to determine, find some biomarkers and create predictive models. So, briefly, let me start with a simple statement. Transcriptomics, it's not just a readout, it's a mechanism sensor. For DILI and NASH, we often face the same problem. Clinical or histological endpoints are relatively late, heterogeneous, and sometimes non-specific. And transcriptomics here can give us a way to observe which biological program or pathways are engaged, up or downregulated, and this often before we see a strong changes in classical biomarkers. So basically, the transcriptomics will help us to capture the early biological process. It will help us to support the interpretability, of the data via the identification of specific programs and not only genes. When we will perform transcriptomics analysis, we will have what we call the DEG, the differentially expressed gene analysis, and we will have some list of genes which are up and down regulated, but this gene list, it's hard to interpret and often unstable. So it is recommended to transform this list of genes into pathways, transcription factor activity or footprint-based signaling to have what we can call units of biology, which make easier to compare the data across experiment, across species. Also, we will talk about this later. So this is particularly important in DILI, for example, where we have different drugs who can trigger different proximal events, but converging into a smaller number of downstream programs. Another particularity of transcriptomics, it's that transcriptomics enable the signature-based comparison across cohorts and model. In both DILI and MASH, we rarely have a single perfect dataset. We have different cohorts, different species, data coming from different platforms, different co-founders, and transcriptomics in one of the methods allowing us to define a signature and ideally transform this signature into a biological program or pathway. Um, and help us to answer different questions. Does these pathways replicate in other datasets, or does it map from a mouse, zebrafish to human, or does it appear in primary hepatocytes and human cell line, for example? So, so much to say on this very um, tiny slide, but transcriptomics, it's a practical bridge towards translation. And it will add value due to the fact that it's a sensible method, give us some mechanistic and informative information that we can transfer across studies. So no, I have clarified a bit why transcriptomics it's valuable uh, in DILI or MASH. The next question could be, okay, what are the signatures we can rely on what we can do directly with this kind of data? So in DILI, first, transcriptomics give us a mechanistical transparency. It will, instead of only observing an endpoint, here we will be able to reveal the mechanism of action and observe which programs are up or downregulated, which pathways are involved after on our observation. So, in other words, transcriptomics help us to move from there is an injury to okay, what mechanism it's likely involved in my injury. The second part, it's okay, the data who come from a transcriptomics could be used as AI predictors because it's provide an high-dimensional fingerprint of the response. And okay, the most important here is not making AI for AI. It's mostly okay. Transcriptomics profile provide the structures inputs to learn patterns, and the AI models love this kind of data, especially when they are anchored in the well-designed dose time or well-created cohorts. So with this kind of data, we are able to build a model that can predict the toxicity patterns or classify mechanism related signatures. Finally, in the daily approach, we can use transcriptomics as a quantitative measurement, or MDTR, for a multidimensional transcriptomic rule. Here, rather than relying on a single marker, we will summarize the response of multiple genes or pathways in order to obtain a robust and quantitative score that can be compared across the different conditions and dataset. Among the different studies we use the transcriptomics in DILI, the classical example is the publication of Zhang and colleagues in 2014. Here, they studies the early response. By data mining, they study the early response of liver to drugs using the TG-Gate dataset, and then identify potential the gene which are involved in this early response. So AGR1, ATF3, for example, to identify this. More recently, Last year, Church and colleague have been explored the blood RNA-seq to distinguish ADL versus other liver injury to show that transcriptomics changes may complement standard biomarkers. They identified 39, 39 or 14, 40, sorry, DEGs across different patients, which suggests the potential for blood-based transcriptomics biomarker to help in the diagnostic and prognostic stratification on KD. For MASH, transcriptomic is useful for more or less the same part, but also for slightly different reasons. It's helped also to resolve where the signal lives and how patient can differ. Using technologies like single cell or spatial transcriptomics, we can we can locate the pathogenic niche, which, for example, which macrophage state, which stellate cells, or which kind of program are activate, and where they are located into the liver. That is essential in the MASH studies due to the fact that this pathology is not uniform and uniform across all the liver. The second support of transcriptomics in the MASH is the patient stratification. Patients can share similar histology, but histological diagnostic, but still fall into distinct molecular clusters, different dominant programs, different immune results, a different metabolic background. So the molecular stratification here become the foundation of the explanation about why some patients progress faster or respond differently than others. Finally, a third point that where the transcriptomics can help in the MASH analysis, it's to connect to connect and identify some predisposition signature using the transcriptome while association studies, or TWIS. Basically, here we will link the genetic regulation of expression to the disease risk of progression. And here the key point and the key idea is that the transcriptomics become the bridge, become the genotypes and the phenotypes, moving beyond a single marker toward the whole program, the whole pathways, whole um, the whole impact of the disease on the liver. So transcriptomics in MASH could be used as as an approach to studies kind of universal mechanistic readout and increase interpretability and support predictive or quantitative framework. In MASH, what is done also, as I said, I talk about the using of transcriptomic, which could be in bulk at the tissue level, or single cell at the single cell level, or even the spatial transcriptomic. Some recent review, which were had published in the Gene last year in 2025, show how to mix the different resolution of transcriptomics to extract very useful and very indicative potential biomarkers or modulated pathways, and mixed with also some microRNA analysis, help for the diagnostic by scoring different genes to determine the grade of the pathology. So using as a single approach, transcriptomics could lead to help us to determine to stratify the patients and predict the potential adverse outcome from just by assessing specific biomarkers. And this is just at the transcriptomics level, bulk, single cell, or spatial. So before going further on the signatures and biomarkers, just a few words about the conceptual approach of transcriptomics. Transcriptomics will capture the systematic differences. This include the biology, but also the tissue composition, some technical effect, and unmeasured confounders. This is why the most important decision when we make a transcriptomic is not the algorithm, but it's the definition of the contrast. What oh yeah what we want to compare to what. It's very important because if we can't say what we want to compare to what, and even the analysis will produce a very strong signal, very strong, a lot of data, we will not be able to know what what the difference we see on the data represents. So this is the classic. This is one of the classic failures on the transcriptomics experimental design. Another one is the batch effect. The batch effect can arise from size, some reagents, some library preparation, personnel, or the sequencing line. So when a batch is correlated with the condition of interest, it can lead to the incorrect biological conclusion. And if it's well documented, if this batch effect is well documented, it's always important to ask the question about, okay, what kind of batch effect I could have on my experiment. The second point, which is very important, is the power rule. Replication and balance design often matter more than the gain of the sequencing depth. What I mean here is to have the robust statistical analysis. It's important and even more important to have enough replicates and well balanced between control and tested condition than having a strong depth of sequencing. So if we want to have to be able to have a signatures that could be generalized across cohort, commodities, platform, it's important to ask the good question and to try to go over this three major failure on the experimental design. As I said, this experimental design is very important, and there's a lot of pitfalls on it. I talk about the batch. I already talk about co-founders, but there is a lot of different variables we could alter the observed transcriptomics signal. Control, time and dose structures, tissue composition, sampling. It's very important to well design our experiment before running a transcriptomics analysis or any kind of analysis, of course, if we want to have a clear signal to be able to answer our specific, our biological questions. And when we answer all these questions, we define our we do, our experimental design. We can go further and ask us the next question about how do we make the differentially expressed genes analysis reliable enough to support signatures and downstream interpretation. Because, as I said in the beginning, we will not just stay at the DG level, but we think it's important to think about how we can use this data and where we will going with this. So the first guild trade here is the replication. Multiple studies show that the same principle, adding biological replicates, substantially improve the power to detect differentially expressed gene while, as I said, increasing the depth of sequencing do not increase this detection. This is important, and the same message appeared in the toxicology dose response setting. There is a recent studies in computational toxicology who show that increasing replicates improve the reproducibility of DAG and even stabilized benchmark dose more than having more depth or more condition. The second part, it's okay, randomized and balance our control and treatment replicates, but it's also important to randomize the position of this batch. I know that in the labs it's not easy to do when we work in the 96 plate, for example. It's not something easy to do, but if we randomized on the plate the position of the different replicates, we will avoid some potential batch effect, not at the lane or sequencing level, but in the plate. I experienced some analysis, transcriptomics analysis, where the first source of variability of the transcriptomics of the data come from the plate position. And actually, we wasn't able to explain why we had this issue, but the fact is when we saw the samples which are analyzed at outliers, we saw that was the same. These samples were in the same location in the plate. So like that, we lost some experimental condition. And with when we have all this information, we will be able to have the strongest differentially expressed genes analysis, allowing us to have this kind of beautiful picture that is the up and down regulated genes according to different samples and condition. But okay, if the gene level, it's give us a lot of information, we should not stay at the gene level, but go from gene level to program or pathways analysis. So I say program because we can encompass on this term different thing. It's, the idea is not new Everyone use transcriptomics to perform this kind of enrichment to Aura or GSEA analysis, just because this analysis were designed precisely to answer the lack of interpretability of the gene level. So the first layer, it's the classic gene set enrichment or overrepresented analysis, which can turn our thousand differentially expressed genes into some smaller number of more or less interpretable pathways. Another level is the activity inference, where here we try to estimate the pathway's activity using downstream footprints rather than only mapping the expression of the pathways. This, for example, could be done with tools like Progeny. Progeny will use the perturbation response genes to infer a signaling activity and explicitly argue the downstream readout which can be which can reflect specific pathways or activity of the expression. And like the pathways analysis, this activity can be this activity analysis is attractive when we want to have a cross-context transfer. The next level is the transcriptional factor activity, just because transcriptional factors often represent the regulation of this program, and for that we can use Darde, which benchmark and integrate a lot of resources to estimating human transcriptional factor activity from the data. So in practice, we didn't need a lot of tools and a lot of disconnected tools, but using this different program analysis, we will be able to compare across different datasets, different cohorts, but also go from the gene level to actionable interpretation on mechanistic hypothesis, cross-cohort comparison, but also prediction model creation or prediction model benchmarking. But this is how the transcriptomics can be used for our analysis. But sometimes it's not enough. Sometimes the single omic approach is not enough. And one reason the field keeps debating biomarkers, it's because sometimes the same phenotype can come from different mechanisms. Two patients can both meet histological definition of MASH, but one be dominated by lipotoxic stress or mitochondrial dysfunction, while another one is driven by an immune activity or fibrosis remodeling. So if we measure only one layer here, only transcriptomics, only proteomics, or just the imaging, we just have the projection of a multifactorial system, and we will see only just the one layer and not all the process. The second issue of this, the single omics limitation, it's the robustness. In running or DD, or when we make some biomarker, when we try to determine potential biomarkers of specific output, it's important to have a very robust one. Any biomarker can work with one subgroup. For example, the non-diabetic or some patient in the specific BMI. But this biomarker could fail in practice. Real-world cohorts will include diabetes, dyslipidemia, or diverse comeditation, demographic variability, and these factors can strongly modulate the molecular readout. So this is this heterogeneity then we can address with a multi-omics approach. So that's why we need this multi-parametric readout to be able to generalize the data we obtain, to generalize the biomarker we identify to adapt it and to adapt it to the real world. So what we can have with the multi-omic approach, so we will have a mechanistic resolution at different level, not only on gene level, because okay, I make a part of the presentation on transcriptomics, but and in transcriptomics we focused mainly on the genes, the differentially expressed genes, and here we can go further. Transcriptomics it's very sensitive to regulatory changes and early responses, but proteins and metabolites often reflects the functional outputs or a physiological state. So it's important to overlap these different layers of information. And another very good point to doing that is that the data it's not redundant, which means we can combine the data from transcriptomics with the data from proteomics and metabolics, lipidomics, to integrate them into integrate them and connect the different dots all over this. So if our goal is to determine biomarkers, we will need to connect what happened in the liver to uh, what we see in the transcriptomics level, proteomics, metabolomics level. And this led us to have something robust. Multiple markers, biomarkers panel can outperform a single analysis because they capture the complementary aspect of the disease and not only one specific layer. So the cross mapping is the key. Yeah. Transcriptomics tell us which programs are activated. Proteomic, it's closer to functional effect and can capture regulation which is not visible at the RNA level, and metabolics and lipidomics often capture the physiological state, especially in MASH. So, in practice, the multi-omic biomarker strategies can be called vertical translation. That's why I put my figure here in the horizontal way. We will first discover some robust pathways, robust program in general in liver, ideally not only in the single gene, but at the different pathways, transcriptional factors or cell type associated to, and when it's possible to support this with proteomics on metabolics evidence, and then translate those information to um, our measurable readout, here the blood, for example, by linking the liver signal to circulating proteins or metabolites, and then validate as a panel, not on a single molecule, but on the panel of biomarkers on independent cohorts or pre-specified endpoint, and then it's very important when we discover new biomarker, it's to benchmark against a standard and the non-invasive test. So the strength of the multiomic approach to detect biomarkers, it's created by the chain of evidence. The chain of evidence where we come that we have. It's came from the mechanism to the measurable readout and to the validated panel. So we are not only see what's happened at one layers, but to integrate as layer as possible for our analysis. This integration, okay, I speak a lot, so I need to be faster, sorry. The multiomics integration is not easy. It's not easy, and it can fail at a lot of level. So it's important to integrate only the data which are comparable. If possible, to use the same. I know it's most of the time it's not possible, but when it's possible to use the same sample to perform the different omics analysis, normalize per modality um, to avoid some the row concatenation or um, don't mix the information between the same samples, meaning okay, we have one biological question to answers. It's not it's not a good thing to have multiple modality into the same sample. For example, we will investigate for the effect of diet, time, and also a time, diet, and a dose into the same samples. No, use common modalities across the different samples. We can use the grade of the pathology. For example, we can use sex. We can use multiple of information to create these common modalities and make the multiomics integration easy. But it's important to think about, okay, what we have what kind of information we have on modalities we have to explain our samples to our data sets and how we can put them all together. And this is where we where the work package 4 opened in the alt running project. The work package 4, it's a work package we will integrate very heterogeneous data who come from different models, in vivo, in vitro models, different species, different readouts. I talk about the transcriptomics for sure, but we also have lipidomics. We have metagenomics also, metabolomics, a lot of heterogeneous information, and it's the first part. It's okay, how to perform this meta-analysis, how to harmonize the analysis with public data, but also all the data generated into the consortium, or combine all the information and what is possible to combine, because for sure all the data can be integrated into the same multi-omics meta-analysis. So it's very important to help us to determine, to go further and determine the potential biomarkers. As I said, it's a multimodal approach, and it's also to develop some specific readouts for the consortium, some reusable reference results, interactive tools for exploration, the program level signatures or grade models determinations, meaning that, okay, maybe we will have some specific models which are more suitable to identify a specific grade of the pathology than another one. So this is one of the aspect of the work package 4. Three example of what we've done so far. It's some first results we obtained with our partners in Ankara and Izmir. So I will not go deep in the information here. The idea is here it's okay. We fed zebrafish with a different diet, high fat diet, and we first analyzed what was the effect of this diet on zebrafish, how we can induce, how these diets induce steatosis in zebrafish liver. And then we also use this diet to induce steatosis on the HepARG cell line, human HepARG cell line. And the next step here, and it's what we are working on actually, it's okay, how we can make or we can project, or we can transfer the results from individual species, zebrafish and human, and make this data comparable. So for that, we work with all the orthology approach, but orthology is fine when we work with the at the gene level. We also work at the program level, yeah, the pathways, metabolic pathways, to find, try to find, okay, is the zebrafish fed with this diet have the similar response at the pathways than the human. So this the first approach, which led us to develop a specific approach to cross species approach. We also start working on the meta-analysis of public available datasets on the MASH. We indexed 9 datasets with the different NAS score to create a big reference datasets and then extract the potential biomarker of each grade of each NAS score using multimodal and multiparametric differentially expressed genes. This is something we work also with work package 3 in the Alteronium. It's okay when we have the approach with the standard classical transcriptomics differentially expressed genes analysis. Sometimes we struggle to determine enough DAGs for to go further on the analysis, on the enrichment, gene scent analysis, or overrepresented analysis. That's why we develop some multiparametric approaches. This is ongoing work also. And another part about how we can use transcriptomics, single cell, and integration of multi-species. It's the MASH-LD Atlas, which is currently we are working on the paper now. It's a cross-species single cell portal mixing data from human, mouse, zebrafish individually, but also all integrated into a big data sets to allow users to perform some differential analysis, pseudo-bulk analysis, enrichment, activity score, and so on. So the idea is to centralize and have these reference data sets for the consumption. Finally, another tools we use for the consortium, it's Toxine. Toxine, it's a toxicogenomics signatures database. It's a reference library of toxicogenomics perturbation signatures based for now only on genes, but it's also compatible with metabolomics and proteomics. The goal is to connect the transcriptome, the programs, the genes, proteins, or metabolites modify after exposure to a compound, to a diet, and linking to pathways, mode of action to understand what's happened and make these signatures comparable each other. Okay, to finish quickly, this is the take-home message. Okay, transcriptomics is a powerful tool. It will capture systematic differences. So the most important decision you have to make when you perform this kind of analysis, it's the contrast. What is compared to what, and which covariate must be controlled. The other part for MASH and DD approach, there is already a non-transcriptomics pattern, but in practice, uh, we can go further and go to the to go to the program level to have some transferable signals, like as I said, the pathways, regularity activity, cell type associated module. Because the same phenotype can arise through different mechanisms, as I said, a single approach, single omics readouts often fail to generalize our biomarkers. So the multiparametric and multi-omic strategy will improve the robustness and the translation of this marker, especially when the endpoint is the biomarker panel. Finally, to have to integrate this data, it's important to align the comparable samples, normalize per modality, control the batch, use appropriate multi-factors method, and use some external validation. And the last point, it's for the work package for running. This work package is focused on the reusable assets by performing a robust meta-analysis in bulk data in multi-method, multi-technologies analytical strategy to increase the sensitivity and to increase the sensitivity for the biomarkers detection. We already developed some tools. We didn't, and we will continue to improve them to make these tools relevant and relevant for the consortium, but not only for the consortium, but for all the community. Okay, sorry, it was a bit long. Thank you for attention. Thank you for the CSM team, but also thank you for the all running consortium for this great adventure. Thank you.
Thank you very much, Thomas. It has been a very illustrative talk. I have a very, well, brief but broad question. What do you think about, well, the topic that is in everywhere right now, the artificial intelligence? How do you think it will impact or is currently impacting in the omics analysis? So what do you think about that? Because I'm a bit techno-pessimist about artificial intelligence in a broad term, but specifically in bioinformatics analysis, it may be a powerful tool, but I would like to know your opinion. Thank you.
Thank you for the question. Okay. As I said, Artificial intelligence, it's a great tool, but making artificial intelligence for intelligence, this have this is a nonsense approach. We still keep in mind that at the end we work on the biological samples. So what we will put on our models, the data input should be highly qualified, highly annotated. So the AI, it's almost the easy part. The real work remains on the experimental design creation, on all the wet lab and all the data documentation, because AI will just do what we want to do. But if we fed our model with uh, garbage data, the output will be garbage. We, it's very powerful tool, but we should keep in mind that, okay, what is the output and is the output of this model? It's having a sense at the biological level. So in the bioinformatic part, uh, I don't like to say that I make some AI approach. I mostly use some okay machine or deep learning, but AI approach it's for me have the connotation of the black box, and if I not understand what is happened in this box, I mean, how can I be able to have a critical overview of the outputs? So AI is good as long as we have the high qualified and very good data in input, as long as we understand what's happened in the box, and as long as we are able to put the results into the in the to put in order the results with biological endpoints and biological relevance.
Thank you. Is there any other questions in the audience?
Yes, if I may. Hi, Thomas. Thank you for your presentation. I just would like to come back to one of your slides where do you talk about vertical translation, where finally you start from tissue and then translate to blood to finally found a biomarker to be validated. And I would like to know whether whether there is already a published paper with this strategy for MASLD, and whether and if any, whether they found some biomarker that outperform the present biomarker that, for example, are used in clinics. For example, the FIB4 biomarker, which is based on ALT, AST, platelets, count, etc. So, is there any published study using this strategy for a MASLD?
Which outperform the standard validation, not in my knowledge. Maybe I missed the paper. It's possible. I saw some papers who talk about this framework with vertical translation, but no one. There is some potential on this framework, but none of the results shown outperform the actual clinical validator. But once again, it's totally possible that I miss a paper on this, and in that case,
Okay.
I should read it.
Okay, yeah. If you have some publication with this strategy, I will be. It would
Yes,
be nice if you can share the reference.
yes, for sure.
Okay, thanks.
There any more questions?
Maybe, sorry, maybe another question. Just, I mean, you in your talk, you talk about Masoldi and Dili. As you know, Masoldi, it's, I mean, a global problem with millions of people that are suffering from this disease. However, for DILI, it's very rare. Most of the DILI cases are very rare. And so there is not so much data that can be used for this kind of approach. Um,
In DD or MASH?
for DILI. DILI, I mean, it's in general, the
Mm.
DILI cases are very rare. And so, I mean, are there any, I mean, it's not you cannot compare. I mean, regarding the bulk of data that you can use for I mean to find a marker, etc. And I just wonder, but I don't know if the question is wise or not. But I wonder whether you can have the same power with only a few data than or than a lot of data, for example, for Maserdi. And I mean, I think that there is less power with DILI, but I don't know if there is some mathematical model that can increase the power of the calculation. I don't know. Maybe it's a silly question, but I just wonder
No, no,
if it is the case.
it's not a silly question because when we perform some meta-analysis, we always want more data. And you're right, in the GD part, I just have a look on the geo and yeah, for sure, the number of data sets available are while smaller than MASH. Um, but for me, it's not an issue if your available data are well representative of the pathology, not focusing on a very specific part and well documented. Because, as I said at the beginning, we have the tendencies that okay, I can integrate more data and more and more and more and more, but more data we integrate, we will also integrate more variability due to the batch effect, due to some misannotations between the data sets. So sometimes less it's better than big.
Mhm.
So I mean, when we saw that Zhang and colleagues perform with the OpenTG gate, which basically was done on the microarray at the time, they was able to determine the early process genes regulation involving in the DILI. DILI. Okay, it's good because the dataset it's pretty huge dataset, but there is less data than for the MASH. But still, the data are standardized, well annotated. So from this part, I mean, it's for me, it's not an issue.
An issue. Okay.
Yeah.
Okay. Thanks.
Lenny have a question.
Yeah. Lenny, go.
Oh, there we go. Can you hear me okay?
Yeah.
Yeah. Hi, Thomas. Thank you for a very nice talk. and a good refresher and lots of interesting stuff that I didn't know about. um, I was just, you mentioned a few times about robustness of data, initially to do either transcriptomics or multiomics. I was just wondering how do we ensure that is what we're, producing? I mean, before the experiment, then we do our, transcriptomics or proteomics analysis or whatever, and how we can ensure we have, robust data post-experiment when we have the data itself. Is there ways to, quality control or, I mean, how do we assess whether the data we have is
Yeah,
high
the quality of the
quality and
data.
robust?
Yeah.
Sorry?
How to assess the quality of your data before having the data and after having the data? Yeah.
Yeah.
Okay. So this could be assessed at the several points. The first one, it's okay, you plan an experiment, so you need to determine the best experimental design as possible to answers a very specific your very specific biological question. Why? Because if from one data set you will try to answers to 10 different questions with 10 sample which correspond to 10 experimental condition, in the statistic way, we will have some issues. That's why you need to have before launching your analysis, it's to have both all the people involved in the data generation and analysis. So having um, people in the wet lab, bioinformatician, having everyone around the table, and okay, we will work on the experimental design. Then it's about how to annotate the data. But if you already have data, then
Yeah,
it's okay.
that's the usual.
Yes, I know. I know, I know.
We don't usually have bioinformaticians co-planning. It
Yes,
just
yes.
doesn't seem to work. It should be, yeah, absolutely.
If you already have your data, of course you have the QC. The quality control will allow you to test to visualize how the sequencing how the sequencing was. Okay, I have enough reads. The replicates seems good. So okay, the wet part was good also. So the QC it's the first part. But then the quality it's also about what questions your data will answer and what the metadata you have on it. So because my worst enemy it's okay, we have a question, but for some samples we didn't have some information about a specific information that we need to answer the biological question. So that means that, okay, so we need to remove this. So the data are robust by having a good having the corresponding experimental design with the biological question, having a good QC and all the metadata associated with your data set.
Okay, so we should book you for future meetings about wet lab experiments.
You have my email, so do not hesitate.
Of course. Is there just a quick another point, maybe related to the AI, there's a paper published just last week, I think it is, Nature Biomedical Engineering on it's large language models
Yeah.
and deep learning and multiomics analysis. Probably saw it. And so is that something that's easy to implement, or is that something you might be looking at in the future, do you think? I mean, some of these large language models are so powerful, they're looking at, complex data sets across different modalities and this is perfect, transcriptomics, proteomics, metabolomics, and all the single cell and spatial stuff.
Yeah, I mean, it's of course it's something we watch. It's something we trying to implement on Ronin also because yes, LLM it's a very powerful tool with some limitation also, and once again, it's um, it's a powerful tool when your data are good in the beginning. I
Yeah.
saw the paper. I didn't read it fully, but the data they use was really standardized, and it was a pretty good quality data. So the
Yeah.
LLM used didn't hallucinate, so that help them uh, to go quick to be quicker and having something very relevant at the end. But there is no new approach, which is the agent LLM or the LLM as a judge. The idea is okay, do not have only one LLM model, but having several LLM model to and let them speak between them to organize
Mhm.
the different level. And this is very powerful in terms of specificity of response, but not also. It's also very powerful because you can integrate external data from each of your agent. To give you an example, I think we are working on in Silicium, and there is some paper about this. You will have one LLM model, one agent. We will rule different sub-agent, but each agent it's linked to a specific databases. And this is the very the part and the difficulty of the agent approach. It's okay. My model cannot ingest all the datasets. I mean, if I give it a list of genes, I will not link my agent with all the cake pathways database. No. The idea is okay. Or it can ingest only the part of the cake pathways of interest for my gene list, and this is the RAG approach, which is no something very where a lot of people struggle to have something performant but also quick in terms of response.
Yeah. Yeah. We need multiple tools to analyze multi-omics, basically.
But it's mostly for the multiomics. It's powerful because with this approach you can have an agent specialized in metabolomics, one of lipidomics, one of proteomics and transcriptomics, and then we can have an agent which ingest the different data sets, um, split it across the different agent. Each agent will not analyze because the analysis will be done before, and will extract the relevant information, compare across the different level, and yeah, give us the potential biomarkers at the different level, which could be related with the translational, the verticality approach between the different methodology techniques.
Okay. So this is work in progress.
Yes.
That's all. It's a very dynamic field, isn't it?
Yes.
Right. Thanks.
Yeah. it's a bit
So,
complicated to keep up to date about this.
yes. Thank you.
Thank you, Remy.
Any more questions? Well, I think it's already time. So thank you again, Thomas, for accepting our invitation, for preparing the talk and giving to us all this, as I said, illustrative and very instructive talk. And Lenie's already applauding. And well, see you in the hard running in-person meetings.
Yeah.
And nothing to add from my side. Thank you very much.
Thank you very much for the invitation, and see you soon.
See you.
Bye
Bye.
bye.
Bye bye.
Bye bye.
Bye bye.